Friday. Today I'm delighted to introduce Dr. Amy Abernethy. Many of you know Amy, but for those of you who don't, she is Chief Medical Officer and Chief Scientific Officer at Flatiron Health, which is a healthcare technology company focused on improving treatment for people with cancer. She's a hematologist, oncologist, and palliative medicine physician, and an internationally recognized cancer researcher with over 400 publications. She's an appointee to the National Academy of Medicine's National Cancer Policy Forum, on the executive board for the Personalized Medicine Coalition, and past president of the American Academy of Hospice and Palliative Medicine. Before she joined Flatiron, she was professor of medicine here at the Duke University School of Medicine. And while she was here, she directed the Center for Learning Healthcare at the DCRI and the Duke Cancer Care Research Program in the Duke Cancer Institute. She's also on the board of directors of Athena Health Incorporated and was featured on TED Med in 2013. Please join me in welcoming Dr. Amy Abernethy.
Thanks. So I have the honor of knowing many of you in the room, and what I thought today we would do is hopefully tell the next chapter in an evolving story that we've been talking about here in the DCRI for, I think, really over a decade. And as we get going, I decided at the very last minute to start off with a story. And so hopefully this all works together because we're going to try it out for the first time. This is intended to be a little bit of my poetic license, as you'll see, but I hope that this helps to set some context. So as I mentioned in the beginning, the topic, or as you saw in the beginning, the topic is real-world evidence. And somebody asked me about 2 or 3 weeks ago, wait a second. First, you're talking about comparative effectiveness research, then we get all caught up in this P-core conversation, and then it's all around big data, and now you show up in today's conversations about real-world evidence. Could you please figure out what it is we're talking about? And, I thought we ought to go back to approximately 1992, 1993, and many of you in this room will remember when David Sackett was having his conversations about clinical epidemiology, predominantly in Canada at McMaster at the time. This led to the Reader's Guide series in the Canadian Medical Association Journal, and then subsequently to a piece in 1993 that was really an editorial or a thought piece in JAMA called Changing the Paradigm of Medicine and Moving Towards a Paradigm of Evidence-Based Medicine. And many of us in the room remember now suddenly being tasked with reading a series of user guides all through the 1990s. And we read our users' guides, and we learned how to read the literature and apply the randomized controlled trial to the care of our patient. We got really adept at systematic review and HTA, and all of this evolving activity led to a series of manuscripts or workshop books at the IOM, first in how to do randomized controlled trials, then how to do observational studies, and how to do systematic reviews. And finally, there sort of seemed to be a statement of, stop this nonsense, let's think about how to do it differently and better approximate research and clinical practice. And that led to a treatise in 2007 called Creating a Learning Healthcare System. That treatise basically said we needed to align culture and incentives and better approximate clinical practice and research so that they're informing each other and we optimize healthcare and also learn within the body of conducting, within the conduct of medicine. 2007 was followed by a really important event. Probably the most important event in the learning healthcare story is this line here. Does anybody know what this line is by any chance? The global financial crisis. So the GFC led to the HITECH Act and ARA following the HITECH Act. And what happened was ultimately the GFC led to the stimulus bill and a key part of the stimulus bill being HITECH. And what had happened was that the IOM book in 2007 said we need a technical infrastructure that allows us to network healthcare providers and allows us to create a system of interoperable, rapidly analyzable data on which we can do this work of accelerating research and optimizing cancer care. That led to a series of activities, many of which are well known in this room. So we have HITECH and ARA. Included in that became an evolving language, which we were all actively involved in around now using this infrastructure to do comparative effectiveness research. And then, by the way, we were a little bit worried about that name, so then we called it patient-centered outcomes research. We saw PCORI. We saw the development of other activities, including the collaboratory and PCORNet, elongation of the activities and the funding for Sentinel and the development of mini-Sentinel, et cetera, and an escalation of activity in that short period from 2007 to 2016 of really starting to figure out what was the core infrastructure and how do we do that work. There was another critical event that happened. So what's the next critical event? This next critical event happened November 8th, I guess, when we had a rapid change in the administration and kind of what's going on in this country. And interestingly, there were, not just the same as when high tech came into place. When high tech was coming in place, it wasn't like the electronic health record and the idea of bringing the electronic health record across all of healthcare was new. In fact, that was well described as a core piece of the infrastructure to getting to a learning healthcare system during this time. It was the accelerating event here that actually put the policy in place that made it happen. Similarly, all during this time, we've had a set of activities that have said, how do we start to do distributed research? How do we figure out how to align incentives? What's the analytic framework? All of that was starting to go on. And then suddenly, we actually have another major event, which rapidly moved along something that was otherwise seemingly dead, 21st century cures. Now we suddenly have a piece of legislation aligned with what's in the PDUFA 6 commitment letter that starts to signal the role of real-world evidence, which essentially was the new form of comparative effectiveness research, PCOR, and now RWE, as well as modernizing clinical trials, surrogate endpoints, et cetera. And so what we see is that these critical events act as catalysts for where we're going. And what we're going to talk about now is real-world evidence and what this might look like with oncology as a use case. And we'll think about how this now lines up to real-world evidence, both retrospective as well as prospective within the pragmatic trials framework, the development of guidances and use cases, and the role of those in ultimately setting up the story for learning healthcare going forward. So that's my version of the story of what's going on right now. So a little bit of backdrop in this conversation around real-world evidence. Definitionally, information derived from healthcare delivered outside the traditional clinical trial setting. In oncology, about 4% of our patients across oncology are involved in clinical trials, and so we generally define real-world evidence as that which is created as a routine part of clinical care or in intentional observational activities outside of formative clinical trials. It includes electronic health records, also claims data. We're talking about registries and patient-generated health data. So sort of a broad range of data types. And one of the challenges here is making sense of it and figuring out how we make it work for us. Ultimately, when we talk about real-world evidence and as we sort of walk through these use cases, the goal here is to complement the traditional clinical trial setting, not to overall replace it, but really make it so that the trials that we're doing then can be complemented with more information that allows for that information to be more generalizable. I'd submit to you that there are many times, as we'll see, when we can use the real-world evidence activities to take the place of clinical trials, but fundamentally, the role of clinical trials of demonstrating efficacy and safety at the beginning is still foundational to what we're doing. And then one of the things that was really important in the December 8th piece in the New England Journal from Rob and others about real-world evidence is that randomization may or may not be a feature. So as we've talked about real-world evidence in oncology, there was a lot of discussion about real-world evidence only being retrospective. And one of the things we've been trying to do in broadening that conversation is reminding ourselves that retrospective to prospective is about frame of reference, and the role of randomization and being able to be intentional about the allocation of patients to one intervention to another is also really an operational activity within the context of this overall point of view. So rapidly moving forward because of, for example, PDUFA 6, 21st Century Cures, but a conversation that's been going on for a while. One of the questions that often gets asked to me is like, oh, okay, wait a second, but that whole big data thing, pull those pieces together for me. Why is this part of the same story? And importantly, when we think about big data, the definitional aspects of big data have been that of enlarging data sets, so volume, often with many different types of source data, so variety, where the data are accumulating off over time, and frankly, that speed that's speeding up, so velocity, and then an accumulating understanding of the quality of those data sets over time, so veracity. So as we think about the definitions of big data, velocity, volume, veracity, and variety, really what we start to get to is a data underpinning from which we can now carve out unique data sets with thoughtful and analytic plans to start to generate evidence. There's an ongoing conversation right now across the US, many of you are involved in terms of what real-world data is as opposed to real-world evidence. For the purposes of the conversation we're going to have today, the relationship between big data and real-world data is that I'm going to call real-world data that information that's collected outside of clinical trials. It's interrelated with big data because much of the big data story right now is happening around the conversation of genomics linked with EHR data, linked with claims data, et cetera. And as we start to think about how these pieces have come together within the oncology community, predominantly led by Friends of Cancer Research, we've been thinking about how do we now start to really show what this means by a series of use cases, where example use cases include confirming benefit of an already approved drug in a new setting, so essentially label expansion, thinking about how to optimize clinical use, so for example, label revisions with new dosing schedules, et cetera, trying to use real-world data to satisfy post-marketing commitments, and also trying to use real-world data to satisfy safety monitoring commitments. And these are the examples right now within oncology we've been working on to try and make this real. Kind of last core piece is I wanted to introduce you to this figure because we're going to come back to it a few times. But this is the way I tend to think about this. So essentially what you've got here is a continuum in time where this big bucket data set here in the middle is, you can imagine, a representative data set representing the amalgamation across a lot of electronic health record data. And when it's being scooped up as a natural observation of what's happening in clinical practice, that's clinical care leading into the electronic health record. And now when the information that's sitting in our EHR systems or other real-world evidence systems is being now thoughtfully processed to be pushed to a clinical trial data set, what we're seeing is the movement now from this information, from the retrospective setting to the prospective setting. So that what you see here is across time, information up until this point in time when there's a consent form and the intentional allocation of the patient to a treatment or something else is retrospective, but there's this moment in time, which could be essentially a second ago, from which the collection of data was all retrospective, and now in this particular setting, second, with consent to do something different to me going forward, those same systems are now collecting information that's allocated in a prospective frame. And as we think about how those data are collected and used, we talk typically about retrospective versus prospective real-world evidence, and the set of use cases tend to sit along that continuum. So, for example, watching within clinical practice to identify patients with rare findings, observing patients that are receiving drug in an off-label setting, real-world follow-up of clinical outcomes, including patients who have previously been allocated to clinical trials, and then ultimately moving into the pragmatic trial setting. And some of the data requirements going forward in order to develop predominantly regulatory use cases, but other use cases require us to think about how do we aggregate across lots and lots of large health systems and other opportunities to aggregate data sets together, especially on the electronic health record side. How do we make sure that this is reproducible, what we're doing, and we can understand provenance and source? We need to think about how do we do data linkage at the patient level. We got to think about how do we make sure endpoints and outcomes are embedded in our data sets. We need to think about how do we have thoughtful analytic plans. This is not just analyzing on the fly, but thoughtful analysis to get to evidence. And critical to all of this is how do you make sure you have thoughtful cohort selection. And so what I thought we would do is talk about the EHR now as a potential way forward in this space, because EHR data, interestingly, can act essentially as an organizing principle or a data backbone. It's very easy to work with because patients tend to have sequential stories across time. We know the individual patient, so at the EHR level, it's highly linkable. The quality can be carefully documented from the EHR and can be systematically improved, as we'll talk about. And you can use an analytic framework that already exists, such as a registry analytic framework, to start to figure out how to work with it. Before I go forward, in the next section, I'm going to talk about some of the key steps we've been thinking about in developing this, but I wanted to sort of stop and see if there are questions or thoughts about all of that, including my crazy story.
Awesome.
Yes. So.
Sorry. In your sort of trajectory slide, you say in the yellow box, real world evidence. Give an example. Are you including things like patient-reported outcomes in real world evidence, or are you only looking at adverse effects that are reported to the feds, or what rolls up under that box?
So we're going to go through 4 example use cases, which will make it really real for you in a few minutes. But I would argue that real-world evidence includes all of the different types of data that can be collected in a natural occurring care outside of the clinical trial setting. So, for example, patient-reported outcomes that are collected outside of the complex clinical trial setting are indeed parts of the real-world evidence data set. Real-world data can be generated from information that is naturally occurring in the EHR. So we're going to look at an example of BRAF-positive lung cancer, for example, in a minute, and looking at the role of BRAF inhibitors in BRAF-positive lung cancer as an example of real-world evidence in that space. Another example is even biosensors, and using biosensors as a way of monitoring activity in order to understand performance status of patients in the real-world setting. Make sense? Yep. Anything else? Hey, how are you?
I'm curious, Amy, what special attributes you think the phrase real world brings to the table here? That paper you cited from the FDA actually was a cautionary one that said, for the most part, real world is an overblown, it's a promise without a delivery.
So I'm going to actually divide that to 2 categories. So on the promise side, I think that the biggest is going to be around generalizability. And so the ability to understand, and I'll show you a case example of generalizability from a collaboration we've got going on with the FDA. On the precautionary side, it's got a lot to do, as we'll talk about it in a minute, with data quality, understanding the source data in a very, very crystal clear way, and then being thoughtful about what questions can be answered with what types of data sets. And my biggest caution in this space right now is that everybody wants it to solve all problems, and it's really, right now there's probably a narrowly defined set of problems that we can use real-world evidence to solve. So I would stick with generalizability for one of the main goals right now. Does that make sense to you, Dan?
Generalizability, there's some inductive problems with regard to defining what you can generalize from and to. I don't want to derail your discussion here because you can get down this rabbit hole pretty far and never come back. But the one thing that happens in the real world is that you pool a lot of very heterogeneous things together and call it the patient population, and it may actually be that the scientific approach of parsing down to a very specific thing to find the treatment signal, for example, is a better way of, if you mix that with all the other stuff, you're just going to obscure something that's really true, and then you're going to say, well, it's not really true in the real world. which is a completely wrong generalization. So it can be a problem to say that this is generalizability writ large.
So I think, and one of the problems is, I think you're absolutely right, we could spend a lot of time in some of the detailed rabbit holes. On balance, the other side of the challenge is that we have so many things that we need to know, for which doing traditional clinical trials to answer all of those questions becomes really almost impossible. And I'll use right now the immuno-oncology drugs as the example, and we're going to kind of get into that a little bit further. Ultimately, with the landscape of change in oncology and what's been happening with the immuno-oncology drugs, just in lung cancer, 50% of patients are receiving these drugs, and we've got a number of problems. So first of all, the patients that were enrolled trials versus the patients who are receiving drugs are dramatically different, number one. And understanding safety in those differing populations as well as efficacies become a huge question. Above and beyond that, all the other drugs that we have that have been studied to date have been studied in a landscape where IO drugs didn't exist. So understanding those drugs and where they line up temporally in terms of which drug should come first, second, third, and the impact in terms of on patients with multiple comorbidities becomes a huge question. And then ultimately, the other thing we're starting to see is drug-drug combinations that didn't exist before either, some of which needs to be looked at prospectively in clinical trials, and some of which at least we can start to understand what are people doing, what does that look like in terms of early signals, and also, frankly, start to understand with payers what payers are paying for within that space. So then one of the challenges right now we've got as we ask the question around IO drugs is literally all of the questions that we need to answer and a lack of ability to get there, yet the answers need to be there today. So we end up with both generalizability as well as temporal ability to get to some information about what's going on. I would agree with you that it would be much nicer to be really systematic about every single question we've had to answer, but we have really no way to get there.
Oncology may be the right place to ask this question in this regard, but I still am listening to Dan closely. If only 4%, 3 or 4% of adults go into clinical trials, cancer trials, and yet you just showed 90% of providers for our oncologists have EHRs. So that would be the test model. How to use a drug, how often is it used, that sort of thing. But Amy, that's gotta be a real spread of reliability and conclusiveness in these data sets across tens of thousands of oncologists.
So this is such an interesting question. I could talk about this particular issue all day long because we now have gotten to the point where we profile essentially every oncologist, every oncology clinic, how they use their EHR with great detail. And I'll give you a very basic example. We understand where clinics that have high quality mortality data versus low quality mortality data are, and we are right now understanding if they've got high quality mortality data, what's their workflows in the clinic that's leading to that high quality mortality data for two reasons. Number one, to understand the implications in terms of the generation of evidence, and number two, to understand how might you learn from what those clinics are doing and pass that information along to improve the workflows in other clinics. And so that you're absolutely right. We've got huge variety. We've got the importance of understanding this all the way down at the ground level with individual providers, patients, and clinics, and then wrapping that back up to figure out how to do all this work. And I would submit that it goes back to the learning healthcare theory. We've got this responsibility of then figuring out how we bring that information back into the clinic in a responsible way. But yes. Actually just realized I've lost my signal, so gotta do that. Any other questions? This is fun. Okay, so one of the things we're gonna talk about now is how we've been solving the problem of preparing EHR data to contribute to real world evidence. Importantly, I'm gonna tell you the Flatiron story. Some of you have already heard that. This is different than how other groups are solving this problem, but at least this is what we've thought has been important in order to do this. Suffice to say, one of the key things that you need to do is aggregate data. Now, that's happened in different ways. So we're aggregating data across health systems, for example. At Flatiron, we have focused on aggregating data across oncology providers, and we do that multiple ways. So one way is we make an electronic health record that's cloud-based. So in that particular instance, which is the largest community oncology electronic health record, all oncologists have their information going into one central common type of database, and that's common. Another is we make software solutions that oncology providers, predominantly in the academic space, but also hospitals, use for a variety of things: quality monitoring, help with clinical trials, precision medicine, and those software solutions then also bring the data in from the electronic health record. These are EHRs that are not one that we make, so like Epic and Cerner, into the same central database. Through those different software solutions, all the data are coming in to one central place. And right now at Flatiron, it's somewhere between 15 to 20% of active cancer patients in the country are coming into one common data set. And we benchmark that against SEER to understand how representative it is of the US cancer population. And it's pretty well aligned with the US cancer population. So we keep up with that at all times. So, these numbers change every month, as you can imagine, but it's about 2,400 cancer clinicians and 1.5 million patients that we're processing at any given time. That's part one, aggregate data. Part two is you need to get everything standardized into a common data model in a very high quality way, and you need to understand the characteristics of data points at the individual data point level. So first, as a reminder about the electronic health record, I always like to think about the EHR as comprised of two sides. This is not exactly true, but schematic-wise, we can think about the left-hand side of this story, which is structured data or digital data feeds. This is the IC9 and ICD-10 codes, or the laboratory data like a CBC. And we've been harmonizing and structuring and putting that into a standard and consistent format for a very long time. And at Flatiron, but also across the country, we all know how to do that. The other part of the EHR, though, is that which resides in the unstructured documents. These are PDFs of medical case notes, discharge summaries, the cardiology visit, the EKG. It's also, for example, the next-gen sequencing test or the condolence card that's the best signal that this patient's died. And the critical variables for doing oncology research reside in both sides, and based on current analyses we've done, about half of the critical variables you need for most oncology analyses actually live on the unstructured side of this. So ultimately, if we think about the structured side, that's classic standardization and normalization. And this is just an example of getting albumin out of an albumin table, getting rid of CSF and urine, and getting to a single concept like serum albumin, and now into a standard set of units. And we do that across all these different EHRs and all these different clinics in a continuous basis for all the structured data. More importantly, though, is the unstructured data processing, pulling in that information on the right-hand side of the figure. And this is an example of why it's so important. So this is a PDL1 report. So PDL1 is an assay that is done in order to understand this particular biomarker in planning for whether or not a patient should receive an immuno-oncology drug. Those of you in oncology in the room, we will know that this is only done about 5% of the time, but that's a different story for a different day. And what you see on this report is there's characteristics about what tissue was collected, there's characteristics around what test was done in the assay that was done, there is the reporting information, and in particular, even this particular lab may have changes in that reporting over time as they learn more about the assay. You really only need a specific set of variables to come out of this report. Importantly, the variables that you need might change over time. So what we do at Flatiron is something that we call technology-enabled abstraction, which I'll tell you more about in just a second, to pull out the current parsimonious core data set of key variables from all these different kinds of documents, but also maintain continuous access to all of the documents over time, so that if the variables that you need change over time, you can always go back and re-abstract new variables over time. Excuse me?
When the lab shifts?
So really interesting question. So a couple of ways. So one thing that we know is we get a lot of feedback from the field. So we're hearing from clinicians, or we hear often from life sciences companies that something's changing in the labs. So that's one way. Another way is we've got a whole bunch of QA checks. So for example, we'll start to see patients where previously we thought that they were PDL1 negative and they're receiving a drug, and now they're positive, and so they kind of hit against our QA flags, and so we go back and check the chart to understand what happened, and then we'll start to see signals that there have been changes over time. So we have, a number of ways these things come in, and then the last way is we do these things that we call spotlight projects. So we'll do deep dive projects where we're asked to do full review, and during those full review projects, we'll actually start to see the changes. So in our technology-enabled abstraction approach, and this is just Flatiron's way of solving this, our argument has been that you still need real people to look at all these different kinds of documents and pull out the data points. And so we've built an environment that allows abstractors who are trained oncology nurses or certified tumor registrars who've also been trained in this overall environment to do this work. We match a particular document with a particular abstractor who's got a task to then fill in the hole in the data set. So if the question is histology for breast cancer, the pathology report is served up to the abstractor who is particularly trained in pathology and breast cancer. He or she knows to look at this document at this point in time and then put that into the data set, into a web interface. And importantly, a number of things there. There's always a connection between what document the abstractor looked at and what information he or she put into the chart. So that would be number one. Number two is that we have the ability to send to 5 abstractors or do anything that we need to do in terms of continuous quality checks and adjudication. And then also in this environment, it's secure and it's essentially an interface that they aren't able to take any documents out or use them separately outside of the security interface. It is easiest way to think about this is we were talking about earlier today. It's kind of like the Amazon's Mechanical Turk model, or it's like an Uber approach to a distributed group of contractors working in a single technology interface to do this work at scale. And we're now doing this across over 100,000 patients per month.
So this is why it's so hard to find good tumor registrars. How much are you paying these folks?
Yeah, we actually probably the number one employer to registrars in the
Jeez.
country. About 500. Yeah. And then the other thing to put in that is a series of controls. And so these are controls that DCRI is very used to thinking about. So there are operational controls, so that all abstracted data, for example, goes through controlled forms, or that there are processes and procedures for every single variable, for every training event, et cetera. There are quality controls, so that there's a process for auditing of the abstraction that's systematic and it is ongoing all the time. And then there's also a review panel for all edge cases. So anything that doesn't make sense to the abstractor first goes to a nurse practitioner review panel and then goes to an oncologist review panel if there's still questions. And there's also system controls such as audit trails. And so one of the important things here is to build the technology that allows that all to happen at scale. And then the next thing you have to do, kind of going back to some of the questions around the quality of what happens in the oncology clinic, is actually to understand really what's the quality of the variables that you can now get out of this to put into your overarching data sets. So for example, here on the left, what we're looking at is what happens if we use structured data only versus if we use this abstraction or curation process to fill in the variables. And here you're looking at histology. So histology, like adenocarcinoma, is only filled in about 1/3 of the time if we looked at structured data coming from across all of our systems. But we can fill it in almost all of the time using curation, because almost every patient has a pathology report in the EHR somewhere. As oncologists, we wouldn't care for them and provide them chemotherapy until we have that information. Similarly, every single variable has its own unique characteristics in terms of reliability, et cetera. So here what you're looking at is different sites of METs. And what you see is that brain METs and lung METs have different inter-abstractor agreements, or CAPAs. And this makes sense when you think about it. So for example, brain METs are more consistently interpreted on a brain MRI or CT scan, and therefore inter-abstractor agreement as to whether or not this is a metastasis and the date that this was assessed is going to be more consistent than a lung MET, where this particular shadow or infiltrate may be interpreted as multiple things and may be hard to characterize as a metastatic focus or not. Does that make sense? And so what you see is every variable has different liability. So once you get to aggregated data that's been processed and that you understand its quality, The next thing you now have is the ability to take those data sets and link them to other key data sets. And so, for example, we take Flatiron data and we link it to external mortality data sets, as I'll show you in just a second. We link it to genomics data sets, claims, et cetera. And that becomes one of the key things that you're able to do once you've processed the EHR is bring in the other data sets to start to add additional characteristics across time. So we track them through multiple ways. So we track drugs through pharmacy records, but we also track them from both the order and administration records directly in the clinic. So we look at all of those. It also depends on what kind of clinic it is. So, and what data sets we have for that individual patient. So we also, for example, have for some patients their claims data as well, so we also track it that way. So it sort of depends on the exact question. We start, though, with the order administration data. And this is an example of one of the linked data sets that we use is mortality. So it's really important within real-world data sets to include endpoints. And in this particular case, if you just pull in mortality data from the electronic health record, you can pull in information from structured data. So some clinics use a data death within the EHR, but not all of them, as we kind of mentioned before. You can look at unstructured notes. So many clinics have, for example, the hospice note that says that this patient's died, so you can fill it in that information that way. And then we have a set of external data sources that we also link in. It turns out that the SSDI, the Social Security Death Index, is decaying across time, but there's now an evolving set of external data sources, such as obituary and funeral home data, et cetera, that you can now start to link in to start to come up with a consensus data death that now you can use as a piece of information within your data set.
This doesn't require patient consent, I imagine, and so what identifiers are you using to do these linkages, and do they differ depending on the data set?
It differs depending on the data set. It's interesting, it's one of the things that we're going through right now. So there's both deterministic and probabilistic matching in here, and it's got to do with the source data sets for the external data sets. So some of them come with, SSDI obviously comes in with Social Security number, which may not be perfect, but it is pretty good, whereas the funeral home and obituary data does not come with that level of information, but comes with name and location. Hey, Eric.
How do you know what you don't know? In other words, how do you estimate the missingness due to patient compliance issues or due to challenges with the utilization of the EHR for information that you would imagine would be important to drive outcomes that you can't? So in other words, do you have a way of estimating the missingness of potential data?
It changes on the variable level, as you can imagine. So this is something that we're thinking a lot about right now, and I'm just going to use outside hospitalization information. So you can imagine that if one of your outcomes of interest is hospitalizations, and you're talking about community oncology, all the hospitalization data that we have is either if the oncologist wrote down this person was in the hospital, or if you have a claims linkage data set in some way to fill in that information. But if the patient is getting care in an academic center, you're going to have hospitalization information that comes from that academic center, but you're going to still be missing the hospital down the road. So we try and go through at a variable level what would be the reasons that we might have missingness and start to understand and compare those different groups against each other, and then try and find, as close as we can find, some golden source data set that will allow us to now do a comparison. So for example, within the hospitalization, we have some really very high quality claims data sets that are pretty good for telling us the complete hospitalization story for a patient, and then we can back our way in to understanding the characteristics for the different source clinics. Does that make sense? And we're able to do that across most variables. One of the issues with mortality data is how do you understand which deaths you're missing? Mortality data is probably the biggest drama in my life at the moment, and it turns out that the National Death Index is our current gold standard, and so we ping against the NDI, but as you know, the NDI is a couple of years old. So that creates problem number one. Problem number two is it turns out we find patients that have passed away that aren't in the NDI. So we're trying to figure out how to deal with the specificity issue there. So we're actually working our way through. And if a person's passed away, we still have the ability to go back and do a fair amount of tracking to see if we think they've passed away, have they really, before we actually say that there's a problem in the golden data set or the golden source. Does that make sense? Probably not going to get to as many examples as I want, but I'm going to at least show you one. So the last piece is organizing data sets for analysis. So the other thing about getting ready and real-world data is actually making sure that the data sets are organized. We do this based on longitudinal journeys of individual patients. So here what you see is non-small cell, and a person gets diagnosed with stage 2 and has a series of events over time before they may pass away from their disease. And the way that we think about this is like clinical trials data sets and populating your data sets in the same way that you might ultimately organize them if it was a clinical trial. So we use structured data to populate some of the data set, and we do so temporally. So for example, pulling in baseline information such as information that would contribute to age or race or insurance, information about therapeutics. So for example, the administration data we were talking about a minute ago is structured data. Filling in parts of the data set using unstructured information and making sure it's thoughtfully aligned. So for example, date of surgery right now, because we can't be sure that we have all the right dates of information for surgery using structured sources is an abstracted data point for us, and that's filled in using abstracted data. Biopsy information, some of the information from pathology is all abstracted. We then link in outside data sets, and we have some linkages that always are in place. So for example, date of death from external mortality data is always in place. We then add in a variety of algorithms that create persistent derived data. So for example, consensus date of death, or one of our biggest derived variables is lines of therapy, which is a big issue in oncology. And then link in outside data sets on a one-off basis when they're available, such as claims data or next-gen sequencing data, to create then large-scale organized real-world data sets that are ready for analysis. And we set them up essentially in, if you sort of think about the table space, that looks very much like the same kinds of data sets that you'd have for clinical trials, but I want to point out one thing. So here, if you're patient A, and we want biomarker data such as EGFR for lung cancer, what we can now do is go all the way back to exactly what was looked at. So it was abstracted by Sue Smith. This was the date that she abstracted it. She was actually looking at this particular document when she did so. She made this particular decision. We know how good Sue is compared to her peers. We know the last time she was trained and the last time she was tested. We know exactly how many people have also abstracted this if it underwent duplicate or triplicate abstraction. And we also have the appropriate audit trails in place. So then this allows us now to get back to all the original questions to make sure if there are outliers or things that we don't understand, and also to make sure that there's the right kind of auditing. And the last point here is, because then things are ready and organized, the next part is you want to always have very systematic analysis plans that go along with this work. And so one of the things that at least we work on a lot is making sure we have a structured approach for all analysis plans that go along with these data sets for anything that we're doing. And kind of all together, what we've found as we've been working on how do we get to the point of real-world evidence that can now start to be used for a series of progressively more high-risk use cases, including regulatory use cases, we can solve the problems such as aggregation and understanding data quality, and we can also make sure that we've got all of the audit trails in place and analysis plans in place that everybody can follow. So kind of with that, I might switch over to some examples to make kind of what Steve was asking very tangible. Before I do so, though, any questions about that? Kind of how we've been solving this.
So, Amy, just from a philosophic perspective, what you just described is extremely personnel intensive. So what's happening on the, or what perspective is Flatiron taking on the front end to minimize the amount of personnel that are required to check, abstract, double check, create audit trails, and do all of that? How do we get to a point where the data coming in is good enough that you can begin to minimize the number of people that have to be hired and sit in cubicles to do all this work?
So I think the first part is to get the overarching kind of approach and infrastructure into place, and then to start automating out different pieces. So, the advantage that we've got at Flatiron, and for those, I didn't actually say this in the beginning, I kind of intentionally almost left it out, which is that Flatiron Health is a health technology company in New York City. We're Google Ventures' biggest investment in healthcare. Probably what makes us different is that we have about 400 employees right now coming from Google and Facebook and Amazon. Daniel here started the drone program at Amazon, and now he's at Flatiron in his next foray. So, Amazon, and then also people coming from the life science industries and people coming from academia, all trying to solve this. So the whole idea of solving it is to first figure out what would it need to look like that we're going to solve to, and then how do we automate out different pieces. We own electronic health record, and we have the ability to do all of this. So that means that from the standpoint of how you now automate out pieces, we've got one machine learning team that's thinking about what are all the places that machine learning can start to wick away work within this system. So can the machine learning help the abstractor get smarter about where exactly to look within the document without creating bias? Can we use machine learning to get better at cohort selection? So some examples. Make the document itself. And that's a huge book of work for us. So that's kind of one example. Another example is how can you get smarter about what you parse to whom? So nurse practitioners and really trained specialists should only do this kind of work because this really is top of their license, and they shouldn't do this. And actually, this stuff we could send to India. So how do you get smarter about what that looks like? And then also, how do you take lessons learned, like what I was talking about with the workflow for mortality data, and start to build new tooling into the electronic health record that starts making it easy within the context of the care of oncology patients to do that work. Does that make sense? No, I think it's like, it is the fundamental question. And I do think that that's the thing that we have to solve for. But the question is, which thing do you do first? And if we waited to solve for all of that, we wouldn't even be sure that getting to the point of having EHR data useful for evidence generation was even a useful thing. Does that make sense? So I'm going to show this example, and then I'll switch. So I'll start with this one. So this is observational data to examine effectiveness of approved agents. And kind of as a reminder, this is prior to randomization. This example is the one I alluded to before. So the BRAF V600 inhibitors have been improved in melanoma, but they have not been approved in non-melanoma BRAF positive cancers. There was a study, it was a basket study published in the New England Journal that looked at the role of BRAF inhibitors across a number of different histologies, so lung and gastric, et cetera. And what that study found was the suggestion that some histologies, BRAF inhibitor does lead to tumor response in a meaningful way, but for other histologies, it does not in the non-melanoma setting. So for example, a BRAF inhibitor for a person who has BRAF positive lung cancer seems to lead to a meaningful tumor response, but for patients who have a BRAF positive gastric cancer, that does not seem to be a consistent scenario. The question was whether or not we can find these rare cases of patients who are BRAF positive and who've received a BRAF inhibitor. Importantly, why might patients with lung cancer get even tested for BRAF status in the first place? It turns out that BRAF is a standard part of most next-generation sequencing panels, and so they're getting tested for BRAF within the context of next-generation sequencing, so it wasn't something that the doctor had to intentionally do. And so what we did was we used, at that time, there were 25,000 patients in the elongating lung cancer cohort to identify all patients who were in the lung cancer cohort that were BRAF positive, predominantly due to next-generation sequencing testing, and whether or not they received a BRAF inhibitor and what happened. Two things about this example. So the first is this over here on the right is just an example of what it takes, or it took us to get to this particular cohort. And we started off with 27,000 patients who were in what we call the Enhanced Data Mart, which is essentially our registry program with lung cancer. And we had to look for a lot of different ways of finding the cases. So we found for any patient who had a history of next-generation sequencing testing, that was one way of finding the cohort. Anybody who had a structured order for a BRAF inhibitor, that was another way. And then anybody where we did free text search and also something called document search in order to find patients. And ultimately had about 1,000 patients who had a suggestion that they had a BRAF mutation. We then found, indeed, a little less than 50. There's a reason I'm not telling you the exact numbers. A little less than 50 patients who were indeed BRAF mutated, about half of whom saw a BRAF inhibitor and half of whom did not. And now we're able to document how these patients have done over time and compare it to the corresponding clinical trial. The reason I'm not telling you exact numbers is that the company who's been funding this work has given us permission to talk about it publicly, but they're still in the final QA steps before we actually get it presented to the FDA. So those are the numbers. And the other thing is that because now you can start to line up these stories across time, what we end up doing is lining up the data set much in the same way I was showing you the structured data sets. And one of the things that we've been working on is being able to document outcomes. So you see here, here's the documentation of the patient's BRAF status. Here's what they received in first-line therapy for lung cancer, second-line therapy. Actually, that was maintenance. And then here's where they progressed and ultimately received now the BRAF inhibitor. And this is the documentation out on the right of their response to the BRAF inhibitor. In this particular case, it was a partial response. But we're able to monitor this for all these patients across time. And one of the things that we're finding right now is the number one use case in oncology isn't what happens for 20,000 patients, but it's actually what's happening for small cohorts of patients that are otherwise really hard to find. The second example that I'm going to show you, since I have a couple minutes, is what has been going on in cooperation with the FDA. So in this particular case, we're now talking about observational data of off-label use and real-world follow-up of clinical outcomes. And this is a partnership that we have with the FDA right now, where they're looking at our lung cancer patients and trying to understand how has there been rapid change in oncology and what's the role of these immuno-oncology agents. So these are drugs, if you've heard about, for example, Keytruda, Opdivo, et cetera. How are these drugs being used and for whom? This is a data set that we provided to the FDA in March. Importantly, we started off with 55,000 patients with lung cancer, confirmed their diagnosis by looking at pathology underneath within the chart before actually allocating them to the cohort. It turns out that of those, there were 23,000 with a confirmed diagnosis of advanced lung cancer and 1,578 who received a PD-1 inhibitor. When we looked at that, what this table is essentially table 1 from the main immuno-oncology studies done in lung cancer. And when we compare the Nivo and Pembro studies versus what we see in the Flatiron data, for patients who were over the age of 75, in the Nivo study, there were 7% of patients in that study with a total N of just around 300. In the Pembro study, it wasn't reported, but the median age was 64. In the Flatiron data set of over 1,600 patients, there were 27% of patients who were over age 75 receiving immuno-oncology drugs and receiving specifically PD-1 inhibitors within the first year of these drugs being approved. And interestingly, that aligns up with what we know of the patients who are actually currently getting treated with lung cancer across the country. That allowed the FDA to start to look at what are the characteristics of patients who are older who are receiving PD-1 inhibitors, and are there any differences in, for example, safety events, mortality, et cetera. This is actually how we've started to look at that. So this is the ability now to layer on dashboards and other visualization tools. So in this particular case, what you see here is that 14% of patients, and this has been of great interest to the FDA, in this particular data set, 14% of patients were receiving PD-1 inhibitors in the first-line setting, although they weren't approved until just a couple months ago for first line. So this was 14% of patients, and then we were able to explore why was that the case. It turns out that the majority of time, interestingly, it's because patients were treated for cure in late stage 3. So we're able to go back and understand why that was actually happening. Then we were able to drill down on this particular group here who got PD-1 inhibitors in the second line setting. And what we see is that when we drill down in that group, the majority of patients are receiving platinum doublets prior to their PD-1 inhibitors, and we're able to now start to understand what's the implication of that order of care in this overall population of patients who are predominantly elderly. So I'm going to stop there. Actually, I want to stop there and then maybe just sort of say a little bit back to where this now kind of gets us as we look forward. So what's happened is we're starting to see that you can generate data sets that start to answer questions like this. These data sets now start to have the characteristics that people need in order to understand quality, as well as understand what any particular data point means in the context of an analysis. And the other thing that this does is create an infrastructure where the next step is to do the obvious. If we've got an infrastructure that allows us to be pretty close to what's going on in the clinic, you can now embed randomization. You can now embed the process of getting patient consent and randomization, as well as if the patient, as well as intentional allocation to one treatment to another, and now start to use these same data sets to serve up and become the data sets for the clinical trial. And so where we are right now is now starting to push these data sets into the clinical trial data set and allow that to become the pragmatic trial data set. And that's sort of where we are in sort of the developmental frame.
Identifying patients and practices that might be eligible for a trial, pragmatic or structured, that this will work to increase that number from 3 to 4% to a higher number?
So it's really interesting that you ask it that way. So here's what we have evidence of right now. And one of the questions that we've had is what's the number you're really trying to increase it to? That's one of the things that we thought a lot about. So we have great evidence right now that we're able to find patients with, for example, complex biomarkers or patients who have rare mutations and rare alterations, and we're able to do that in near real time. So for example, patients with MET alterations, we're able to find those patients and do one of two things: have them ready to go in a clinical trial when a clinical trial becomes available for them, or identify them and mark them as eligible. And because we're already in the clinical workflow, we can signal back to the doctor in real time, this patient's potentially eligible for this trial. So we've been able to do that. And the places where we've been able to short circuit is identify patients for studies which usually take 4 or 5 years to enroll, and show here's the 40 patients that are already ready to go. What we have not done yet, but really mostly because we actually haven't tested it yet, is now ask the question, if the baseline rate in this particular clinic is 4% or 25%, depending on the clinic, and now we start to add in the following things: better patient identification, new workflow solutions to make it easier to do clinical trials in the clinic, the ability to use real-world evidence data sets to make it faster and smarter. Do all of those things now make it both easier to find patients and actually more palatable to have patients on clinical trial in your clinic, and do you see rates of accrual go up? Does that make sense? And that's what we're doing right now.
And this is a critical need in oncology, since we have far too many trials in question than patients on study.
Well, the other thing that we're able to do right now is, because we can see 20% of patients across the country, we can do two things with that. We can actually say these are the sites that are seeing predominantly GI oncology, and we know that they've got 30 second-line patients coming in on average per year. So with that, you can do a lot of things. You can actually say, here's the sites you should target for your trial, one. You can say, by the way, when they fill out that form and they tell you they have 60 patients, they really don't. They only are going to have 30, of which only 20% are going to go on trial. And you can also, with that, basically say, here's going to be the portfolio of trials best for this particular site. So that's one way that you can handle it. The other thing that you can now do is start to say, here's a smart portfolio nationally for this particular development program or this particular. So that's the other kind of direction that we're going.
So along those lines, then, pulling is great, but how do you push? How do you take this data that that gets aggregated and then push it back down to individual clinics or cancer centers in a meaningful way that they can take the data and actually track back to a cohort of potential patients? That's way more complicated.
Yes, so that's, I honestly think that this is going to be the next wave. If I, kind of go back to the story that I told in the beginning, we have where we are right now in terms of the development of real-world evidence and everybody talking about it. Now the next task is actually figuring out what does it really mean? What kind of questions can we answer reliably? How do we do this work safely? And then the next step that comes right after that is saying, how do we push this data back into the clinic? I don't think we actually have to wait all the way until we've learned how to answer 50 questions to be able to figure out how do we push back into the clinic. There's some safe things we can do right now. So, for example, we can safely say, here are patients that you have with unusual alterations. Have you considered clinical trials? So one thing you can easily do is that kind of work. You can easily start to push, for example, pathways information and coded information that goes along with guidelines back into the clinic and do that within your EHR. And then you can start to say, here is data that's coming from an aggregated format from across the country that you might want to think about. But getting to the point where now you say, here's your 30 patients, and here's how you should think about them, I think actually takes us to go through all these steps before we do that. Does that make sense?
Yeah, I think it's a lot easier to tell the FDA that there is a large cohort of patients who have a BRAF mutation who have gotten some treatment off-label and it's working or it's not working. And you should go forward from a regulatory perspective on that. That's a no-brainer to me. But trying to get it back into the real world in real time is the tougher part.
I would argue that we have an ethical imperative to actually get this really right before we start doing that, because if we just start pushing a whole bunch of data back into the clinic without being sure that we understand how to analyze it, being sure that you know what's credible to generate from it versus what's not, you're going to get patients and doctors into a lot of trouble. Other questions? Dan, I'm curious now. Tell me more. You still think I'm crazy?
I never
say
thought
yes,
you were.
by the way, that's usual.
I never thought you were crazy. I have a question, and it's not really along the lines of what you've just been discussing, but it goes back to some of the data issues, which relates to something I've been doing with FDA recently, and that is whether or not your data extraction you're using a lot of genomic and molecular tests to classify patients, fairly sophisticated, complicated laboratory tests that in many cases are done by the individual center, or in some cases they're sent out to a company like Foundation Medicine. But in some cases there are hundreds and hundreds of different versions of the same test across the country, done by different laboratories, and nobody knows how they agree. And there was one paper that just came out in JAMA Oncology, a letter actually of 8 patients where they studied Foundation Medicine and one other assay, I think, and found a 20% agreement in the markers and 25% concordance in the recommended chemotherapy. And so that seems like a fundamental sort of bit of rot in the foundation that's going to prevent figuring out some of these more sophisticated and important things. If you can't rely upon the way you've classified the patient, then figuring out the relationship between the patient's genotype, phenotype, and therapy is going to be almost impossible.
So I was pointing to Marta because I was like, we were just talking about this last night in the car, and it has implications all over. So it has implications for how we set up the data models. It's got implications for how we think about what analyses you can do, et cetera. And it has been something that we think a lot about. Some of it it goes back to, for example, what we were talking about a few minutes ago with even the variant call files change over time, right? Like as you start to come up with more sophisticated ways of analyzing the genomic data, the VCFs change and then the alterations and their actions change. Similarly, if you sort of just think about, we were talking about ER/PR status, which is just wrought with drama. And it's different all across the country as a series of laboratory developed tests. I've just kind of reiterated the rot that you've just described. So first of all, I would submit to you, at the end of the day, so we still have to take care of patients, and that's what we've been doing. And so we start off with the premise that these are the tests in the current interpretation for which doctors are making decisions on that at least gives us this particular line in the sand. That's part one. Part two is that we should never think that what was generated at that particular line in the sand is now dogma from that point forward. So we've even been thinking or had internal conversations about, do you require some set of thoughtful updated analytic plans that happen sequentially over time to see are there changes because of changes in what we understand about the underlying data, changes in what we understand about the biology that would change either the analysis and the results or the interpretation, and do you actually bake that into the process over time? And then the other thing is, as you think about the data model, do you create a parsimonious data model on one side, where, for example, if we just use PD-L1 expression assays, it's PD-L1 expression assay and the result that was given on this test to this doctor.
That's the worst possible example.
Well, that was the reason why I picked that one, because it's just, yeah.
Unpredictable and the most variable test imaginable.
And the cuts are all over the place, right? So sometimes it's 5%, sometimes it's 50%. So we're just gonna, what if we just start with the parsimonious data model that says PD-L1 was tested on this date using this assay, and this was the positive or the negative. And then instead of adding everything to the data model, actually check across time with deep dive reviews of the assays and the reports and how that changes to understand how that works across time. So the kind of what we're really trying to say is we can't not try and generate evidence, but we actually can't put our head in the sand that the answers that we got today are now the answers that need to persist across time.
If you don't actually have information that may be turned in a slightly different way, can shine a spotlight on something that I think within the professions of laboratory medicine and perhaps to some degree oncology as well, hasn't, and other disciplines, but those are the ones that are most intensive in this area, it hasn't been grappled with, I think, completely. The implications of some of these things and the current policy approaches that have been taken have created these dilemmas for us figuring things out that make it takes a very complex, horribly complex problem and makes it vastly even more complex to figure out, and that's not so helpful. But I wonder if you don't have some of the information that could be used to shine the light on this problem and get people thinking about how to
Here's
see
the
outcome.
assay environment I would love to hand to you to analyze. So I'll just use HER2 as the example. We know all the HER2 tests, right? We know all the patients who received trastuzumab or trastuzumab-based therapies, and we know their outcomes. So we should be able to interrelate the actual test to predicting therapy and outcome and be able to start to understand if, for example, at least there's groupings of different kinds of laboratories that are better at predicting which patients should receive HER2-based therapy versus those who shouldn't. And interestingly, I use HER2 because all these LDTs, for the standpoint of laboratory developed tests, just all over the place once you get to that point. And yet there's no national approach to how we actually make sense of this. So we have data sets and we're looking for academic partners.
Using outcome data, which would be response to therapy, to go back and say, does this test actually predict anything? Or even more specifically, does this particular test, compared to other tests, more accurately predict response or not, right? So you're basically reverse engineering.
That's exactly the environment.
Okay.
That's the, we right now, we just handed Yale, and I'm not embarrassed to say, just gave Yale all of our NGS data and said, here, 30,000 patients with lung cancer, fully curated, here are all the NGS tests that they've received, and then there's all the patients who've had NGS testing, and there's all those who haven't, and there's at least a 6 to 8 to 1 propensity score match between the two populations. So, yeah, it'd be pretty cool. Any other questions? First of all, thank you guys for staying late.